Hello students, so in this lecture we will be discussing about light tissue interactions. Now as in normal material, a non-living material, the living or the tissue when getting interacted light, it undergoes the following phenomena of scattering, absorption, uh, reflection, refraction and transmission. Now we'll first start with the scattering phenomena. Now, the scattering, as we all know, can be subdivided into either elastic scattering or inelastic scattering. Now, elastic scattering again, and this goes for the living tissues as well. In elastic scattering, there are two types of scattering. One is the Rayleigh scattering and one is the Mie scattering. Now, the Rayleigh scattering is mainly for small particles, where the particle size is much less than the wavelength of the light used, whereas in Mie scattering, the particle size is much larger and is also spherical in nature. Okay, now like here, the particle size is larger and spherical in nature, and if we compare it with the ones that goes on in the living tissues, it is mainly observed in the tissue where there are skin involved, human skin, human brain, the muscle, the bone, and any other soft tissue like your liver, or kidney. Liver, kidney, spleen, all these have soft tissues in it. So here the light scattering will be following this Mie scattering pattern, and it is this scattering has got applications in optical coherence tomography OCT, confocal microscopy, laser induced breakdown uh, spectroscopy, and photodynamic therapy, and the tissue diagnosis. I mean, finding out whether the tissue is cancerous or not. So in all these cases, this Mie scattering or scattering from very large particles in the tissue is applied. Okay, now coming to the theory. Of this Mie scattering, the intensity varies with the angle of scattering, as you can see, I theta, and I0 is the incident intensity, and so the Mie scattering intensity has got units of watts per meter square per sternian, that is the solid angle unit, whereas incident intensity, the light that is being supplied, has got units of watts per meter square. Here there is one more, there are three more terms in fact. This alpha here represents the radius of the spherical particle, whereas r represents the distance from the center of the spherical particle. And lastly, you have another quantity, this is the scattering efficiency. And the scattering efficiency again is equal to 8 by 3 alpha by lambda to the power 4. So Mie scattering, although it comes under the banner of elastic scattering, there are some non-elastic parts associated with Mie scattering as well, because of this large size of the radius, large size of the tissue particles. Now the cross section is again given as by a square alpha square, that is the radius of the spherical particle, and the scattering efficiency. Okay. Now the tissue character, to characterize a tissue by means of light, the parameters that are used, that are considered, are mainly the scattering coefficient, then the absorption coefficient, the refractive index, and the fiber orientation and density. So in terms of scattering coefficient, which these coefficients, both of them have value inverse of length. So it clearly indicates that if we have a tissue or any substance, it is how much depth, if the coefficient is high, that means the scattering is taking place from a shallow depth. If the coefficient is, that's why this inverse centimeter unit, theoretically or logically you can think of that. Now if you compare with the different, some of the tissues, like in skin you have the scattering coefficient between 100 to 200, whereas the absorption coefficient of much higher value. So if you take the ratio of these two, absorption coefficient versus scattering coefficient, you will be qualitatively be able to uh, say that which material scatters more and absorbs, how much is the ratio of absorption versus scattering. Like in muscle, the ratio goes a little down. I mean, not that too much down, little down. 50 to 100, whereas 1 to 5. Similarly, when the material gets harder, the ratio increases, and when it gets smaller, softer, the ratio again decreases. Now, the refractive index, since there is a lot of water content in these tissues, so they remain around the value of water, 1.5. Now, compared to this Mie scattering, if we take Rayleigh scattering is the one which is why we observe in the normal, in the world we observe the rainbow colors and all. So the Rayleigh scattering is basically when the particle size is much less than the wavelength of the light being used, and also there is an extra term being added apart from the change in the intensity expression, numerical changes. There is one more extra term being added. So in the case of Mie scattering, we have seen that the radius of this large spherical particle was very important. However, in this case, since there is an electromagnetic radiation, so this electromagnetic radiation induces some polarization in the molecule, in the sample, and the polarizability, how much it is getting polarized, it is a factor in the intensity, the Rayleigh intensity. Now since the particles are small, is usually the scattering is elastic in nature, so the polarization plays a part in it, and also there is an angular factor to it. So you can see the Rayleigh scattering. Then coming to the the ones which are inelastic in nature, that means where uh, you can find the wavelength of the light, scattered light, to get changed. That is Brillouin zone scattering, which is mainly due to formation of acoustic phonons. And you have the very good old Raman scattering, where there is a vibrational excitation being induced into the outward going radiation. I will take up these later in details. For now, let us try to understand that when the light interacts with the tissues, the following phenomena can take place inside the tissue. The first one is known as autofluorescence. Autofluorescence is the phenomena where on being radiated with light, the tissue starts to emit light. So there are natural light emitters. Examples being collagen is a natural light emitter. Now, so apart from that, a light interaction also produces photochemspheres. That means chemical reactions start to take place. There are also light uh, causing some thermal effects in the tissues. Now, and apart from that, there is ablation taking place. Ablation is a term used when the cellular structure breaks down. So we can have two different types of ablation. One is the photoablation, where the light is directly involved in breaking the cell structure, and the other one is called plasma ablation, where due to high intensity short pulses of light, there is a creation of plasma. Plasma is where the electrons and the anistate neutrals, they all stay in equilibrium. Now this plasma causes the breakdown of the cells. So ablation is another property. And lastly, we have photodisruption. Photodisruption is equivalent to a mechanical cutting of the tissues. Now this mechanical cutting will take place if on generation of some shock waves. Now these shock waves are analogous to using hacks to using a mechanical cutter to cut the tissues. So these are the different properties going on. Okay, now moving on to the absorption and the emission that takes place in fluorescent in tissues or any other molecule. Now often it has been observed that the absorption frequency or the absorption wavelength is not equal to the emission wavelength. There is a difference between these two. That means the wavelength at which the light gets absorbed in a material and the wavelength by which the same material emits the light, they are different. And this difference in wavelength is known as Stokes shift. Now, the Stokes shift can occur mainly due to the difference in quantum efficiency, energy level spacing, and due to some internal processes going on in the tissues. We are talking about tissues right now. Now, first one is there's a phenomena of fluorescence and phosphorescence, FL and pH. These two are although little different. Phosphorescence is the one where there is some mm, uh, crossing of intersystems. I mean the electrons, they go to an excited state which is normally forbidden, and they take longer time for to get emitted. They take a lot of time to emit the light. That's where you can see the light glow. Now, apart from that, in case of fluorescence also, there are some internal conversions taking place. Like here you can see the light has been absorbed up to this level, so this corresponds to the absorption of light. However, due to some internal conversions, the electrons have gone to a low energy state. So these internal conversions are mainly the ones we discussed right now, that is ablation, disruption. So this one, these are the different types of processes that is taking place due to interaction with light, which is causing these internal conversions to take place, or also sometimes this one. And so we have a different stroke shift. Now this process can be quantified by fluorescence quantum yield. Fluorescence quantum yield is the ratio of two parameters, as you can see, gamma and gamma plus KNR. It is represented by Q, and sometimes it is also represented by phi. So this, what is gamma? And gamma is the radiative, both of them are rate constants. Gamma is also a rate constant, KNR is also a rate constant. The difference between them is that the gamma is the radiative rate constant, and it represents emission of photons, whereas K and R is the non-radiative rate constants, and it represents the relaxation process. That means this process, due to the interaction, the chemical and the physical processes taking place due to light interactions. So that is why this value comes into the picture. Now if the value of phi is equal to 1, that means gamma is much greater than k naught, so it indicates that there has been a minimum amount of interaction causing chemical and physical process in the tissue, and the radiation is mainly radiative, and there is phonon, but there is not much of an energy difference between the absorbed and the emission. However, if the internal processes they increase, automatically this y value becomes less than 1. Now there is a lifetime of the fluorescence process, and it's called fluorophore life, for how much this will be there. Now this is what increases as the internal crossing takes place. That means the phosphorescence, although it is called fluorophore life, but it also values for phosphorescence. It is much higher than fluorescence. And also in terms of difference between absorption and emission, the time between absorption and emission is in the range of nanosecond, and different fluorophores or light emitting materials will have different types of lifetimes. Like in the case of rhodamine, the lifetime of fluorophore lifetime is 3.8 nanoseconds. Next is biopolymers. Biopolymers is nothing but the polymers which are produced by living organisms like proteins, polysaccharides, and nucleic acids, so on and so forth. Now, these polymers are also called light absorbing chromophores, some of them. So one light absorbing chromophore means when on absorption of light there is a series of chemical processes going on, and these series of chemical processes, which is preceded by a primary step, And these series of chemical processes, they produce intermediates in each and every step. And subsequently, when there are also chemical processes where there is no light involvement, so they're called dark processes. Now, the interesting fact about these is that they, even though the processes are going on, they are cyclic in nature. They ultimately produce the polymer, biopolymer from which it started. And so, one of the examples of this is called retinal or retinaldehyde. It is the biopolymer found in our eyes, and it is the basis of the vision. Okay, now this retinal is composed of retinaldehyde, which is 11 cis retinal, considering its structure, and there is an aldehyde and amine group, and when they are bound to a protein called opsin. And so ultimately what is left is rhodopsin. So an 11 cis retinal being attached to an opsin protein forms the rhodopsin. And now this 11 cis retinal has two asymmetric states, the 11 cis and trans. Now when light falls on this 11 cis retinal in the rod cells, it converts to trans. Now what is happening is that, see here, this is the structure of the cis retinal. The black dot represents the CH3. So when light falls on this structure, this region, it moves, straightens up, and this is the trans retinal. So this is what takes place. This rhodopsin found in the rod cell, it is sensitive to blue-green light, which is around 500 nanometer, and this rhodopsin, this process of straightening leads to it activates a polymer complex called transducin. Now, this transducin again breaks down unseen monosphere, which is a nucleotide and is an RNA monomer. Now this drop in GMP, so since it is breaking down, there will be drop in the concentration of GMP. This drop in GMP leads to a close of the ions, the sodium and the calcium ions inside the rod cell. These ions, because these are the tissues which are responsible for the tracks of the Na plus and Cn plus ions. So when these are breaking down, so automatically the channels get closed. So ions now can't go inside the rod cells, hence there is a potential negative charge being created. So this negative charge causes this rod cell, which is basically a membrane, the potential increases of this rod cell, like from minus 40 to minus 60 millivolt. And due to this change in potential, there is electrical signals being created, and lastly they are transmitted to the brain through the optic nerve. So this is how we see images. So there is a lot of chemical and physical processes involved in the vision. So this is biosensing. Our body is the best sensor, so we can see how complex is our body. It does the following chemical and physical functions to make an electrical signal transmit to our brain to the optical nerve, and it is a continuous process. It goes on. Whenever we are observing a thin, it's not that we are observing at any particular lambda, we are observing different colors. So automatically the lambda changes every time. As the lambda changes, the whole process gets changed. So the electrical signals transmitted and each and every nanosecond is different. So you can understand how sophisticated our the human body sensing system is. Okay, that's it for today.